Hello, everybody. Welcome to the United Leukodystrophy Foundation's virtual family conference session on MLC, presented by Dr. Min, Dr. Vander Knop, Sven Kirst, Quinty Biesling, and Emma Paschier. This session is being recorded and will be posted for viewing online in early July. Thank you so much for joining us. Now I will turn it over to Dr. Min. Thank you.
Thank you very much for the introduction, Tara. So let me share my screen with you so that you can see my presentation. And let me take it away. I hope everybody can see the presentation and can hear me well. Before I start, I would really like to thank the ULF for giving us the opportunity to talk to you today about MLC and about the research that we are doing, where we try to understand the disease, of course, ultimately because we really would like to develop therapy for this disease, which is currently not curable at all. So advance my slides. So in today's session, I'm going to start off with a short introduction about clinical characteristics of MLC. Then I'm going to talk about ions, water, and electrical brain activity, mainly because we think that this is the process or the balance that is disrupted in MLC. Then three of the researchers that work in our group on understanding MLC and understanding what is happening in the brain of MLC patients by doing fundamental neuroscience research are going to talk. These are Quinty Biesling, Emma Paschier, and Sven Kirst. And so they're going to talk about their specific research. And then after that, I will briefly discuss therapy development and what kind of lines we are thinking along. I will briefly discuss gene therapy because, of course, that is something that you probably hear a lot about. And then I'm going to talk about potential other therapeutic approaches, although I should say they are far in the future. I will end with a little piece about how you might be able to help us in our research. To start off with, megalencephalic leukoencephalopathy with subcortical cysts, or MLC for short, is a leukodystrophy with quite an early onset. And so what we see is that children who have MLC actually in their first year of life are mainly coming to the clinic because they have a very large head. So the microcephaly is very pronounced. You can see the normal head circumference curves for kids here. And you see that within the first year of life already, MLC kids are quite far above that head circumference curve. Then in the rest of their lives, these patients often are experiencing epileptic seizures. Not everybody, but quite a few. Cognitive disability can be part of the disease. There is often pronounced motor impairment. And as I said earlier, there is at this point no treatment for the disease. Now, MLC was described for the first time by Mario van der Knaap, who is also here today in 1995, and simultaneously in India by Professor Singhal. And after the discovery of the disease, the search for the disease-associated genes started. And so in 2001, this led to the discovery of the first MLC-causing associated gene, which was called MLC1, very appropriately. And in about 76% of the patients, we find that they have recessive mutations in MLC1, which means that both of the copies of the MLC1 gene that everybody carries are damaged in these patients. But this was not all patients, of course, so the search continued until a second MLC-associated gene was found some years later. And this gene was called GlialCAM. It was also known as HEPACAM because it had something to do with liver cancer before it was discovered that it's linked to MLC. And so in patients that have HEPACAM mutations or GlialCAM mutations, we actually find two different types of mutations. So about 7% of these patients also have recessive mutations in GlialCAM, meaning that both copies of the GlialCAM gene are defective. But in about 15% of the GlialCAM patients, we find that they have dominant mutations. So these are very specific mutations where only one of the copies of the gene is affected. The other copy actually is normal, but still this leads to a disease. And this actually also leads to quite an interesting difference in the types of MLC we see. And so patients who have recessive MLC1 or GlialCAM mutations show classical MLC, and that is these patients develop macrocephaly in their first year of life. Then they have a typical or mildly delayed early development. Epilepsy is quite common in these patients, so about 60 to 70% of the patients develop epilepsy. There is slow motor deterioration, and often these patients become wheelchair dependent in their teens. And then later there might be mild intellectual disability. But it should be said also that variability in the disease severity is present. So there is a very large variability in how patients are affected. And for these classical MLC patients, because both genes have to be affected, often the parents, or actually always the parents, are unaffected. Here you can see an MRI scan of such a classical MLC patient. At 4.5 months, you already see that the white matter, which should actually be black in a typical MRI, as you see here, actually is very bright, and it's also quite profoundly swollen. And this actually stays the same through life. Here you can see the same patient at 42 years. What you see is that there is actually some atrophy happening. Otherwise, there's not much change. And you have these subcortical cysts. So these are these fluid-filled cysts that are in the brain. They are also always there at all ages. Now, when we talk about the dominant GLIOCAM mutation, so these are the patients that have only one defective version of the GLIOCAM gene with a very specific mutation. These patients are in their first year of life indistinguishable from classical MLC patients in that they develop macrocephaly. The MRI looks very similar. They are often also mildly delayed to normal in their early development. Epilepsy is a bit less common in these patients. And then from the second or third year of life on, we see a remarkable clinical improvement. And so there is often no motor decline. The macrocephaly persists in about half of the patients, but in about the other half, actually the head circumference normalizes. And in this patient group, we find that intellectual disability or autism is quite common, although that's not so common in classical MLC. And because of the dominant inheritance, often one of the two parents had a transient macrocephaly as a child, although this doesn't have to have been diagnosed as MLC at the time. If we look at the MRI of these remitting MLC patients, we see here a patient at 9 months looking very similar to the classical MLC, but then at 8 years, the MRI from the same patient shows this striking normalization. Here's another patient at 11 months, and at 5 years, again, you see this striking normalization where the MRI is actually almost indistinguishable from a typical MRI. Now, after identifying the disease-associated genes, of course, sort of detective work started to figure out what is going on in the brains of these patients and why do they develop this disease. And so one of the things that is a very good first step in such research is to figure out where the proteins that are coded for by these two genes can be found in the brain. And so our brains, of course, consist of numerous different cell types. And when you think of brains, often you think of neurons. Neurons are the cells in our brain that make electrical activity. Their electrical activity and communication with each other forms our thoughts. They control our muscles. They control our behavior. But actually, if we look at the amount of expression of the MLC genes in neurons, we find that they actually don't express these genes. So the main culprits don't seem to be neurons. Instead, we find a lot of expression of MLC1 and GLIOCAM in so-called astrocytes. And astrocytes are a cell type that is also very abundant in the brain. We didn't know so much about these astrocytes, but our knowledge of them has grown tremendously in the last 20, 30 years. And astrocytes are a type of glial cell. So what you can see in this drawing here you can see one of these neurons with its elaborate processes with which the electrical signals are sent through the brain. But glial cells, their name derives from the Greek word of glue. They were thought to be sort of the glue that holds the brain together, but they turn out to have very specialized functions. I'm only going to talk about two types of glial cells, these astrocytes and the oligodendrocytes. And let's start with the oligodendrocytes. So they are the cell, you see one of them in purple here. They are cells that actually form sort of insulating wrapping that wraps around the processes of neurons. And by that, they make the signal transduction by neurons, so the communication between neurons much faster. And our white matter contains a lot of these oligodendrocytes that are wrapping around the long processes of neurons. And then the second type of glial cell, so I already mentioned them, that we talk about today are the astrocytes. And here you see one in green. They derive their name from the fact that they are star-shaped, so they have these very nice star-like appearance. And they form very elaborate contacts with neurons, also with oligodendrocytes, although that's not shown in this picture, and also with blood vessels in the brain. And their tasks are thought to include energy supply to neurons because they contact the bloodstream. They can take nutrients from the blood and supply them to other cells in the brain. They can actually modulate neural signal transmission. So because of their contact here, they can tune the way in which neurons talk to each other. But the function that for our talk today is most important is that they control the balance of ions and water in the brain. Now, what do I mean by that? Our brains, of course, contain all these cells, but our brains also, for a very large part, consist of water. And in that water, we have diluted salts, or ions, as we call them. And these are charged particles, so they can have a positive charge or a negative charge. And because our brain is an electrical organ, this electricity is generated by the movement of these ions into the cells or out of the cells. So here I've illustrated how that happens. What you see here in gray is sort of the cell outer wall, the membrane. And these different colored things that I've drawn in are channels. And these channels can open and close. And when they open, ions can flow through them. And here we see a channel, for example, that very selectively lets potassium ions or K+ ions flow through. Here there's a chloride channel. Here there is a sodium channel. Sodium is Na+. And here we see a water channel. And this movement of ions and water is crucial for our brain functioning, but it's also crucial to tune it, because if it gets detuned, electrical activity gets disrupted. And you can also imagine that if the water fluxes in our brain get detuned and the brain would swell up, this would be quite dangerous because, of course, our skull hampers the swelling of the brain. And so this is where the astrocytes come in. The astrocytes sit here in between the neurons. They are sort of sensing the amount of ions and water. They can actually take up ions and water, and they can balance out
Is everybody frozen?
I was for a bit, yeah.
Okay.
Me too.
Did we lose Dr. Min?
Yeah, but I think he probably will be right back.
Okay.
This is the downside of video meetings, eh?
Yeah. Okay, he says he's almost back.
Okay, I think I'm back.
Yes.
Yeah.
Beautiful.
Oh, I sincerely apologize. My internet suddenly crashed, which is of course what can happen in these kinds of meetings, which doesn't happen in real life meetings normally. But luckily everything seems to be started up again. So can everybody hear me again?
Yes.
And can somebody remind me what was the point at which I was lost?
You,
So you were talking
may-
about slide 11.
Okay. So that's basically the one before this.
Yes.
Okay. I was talking about ions and water in the brain.
Yeah, you were at
And
this
I was-
slide.
Okay, good. So, yeah, my apologies. Electrical activity in the brain is generated because ions, so positively or negatively charged salts, flow into and out of cells. So here you can see schematically the inside of a cell and the outside of a cell, and the gray part here is the what we call the membrane. That's sort of the outer shell that encapsulates the cell. And in this membrane we can find channels that allow, for example, different ions to flow out of or into the cell. And so electrical activity in our brain is generated because of this ion movement. Now there's also channels there that allow water to flow into or out of cells. And that's very important in the brain because, of course, you can imagine because of the hard skull surrounding our brain, there is no room for the brain to swell. So this water and ion flow has to be perfectly balanced. And that's where astrocytes come into play. They are perfectly poised between neurons and blood vessels and other cells so that they can balance flows of ion and water when they get disrupted. And now I'm going to highlight one scenario where this would happen. So when neurons become very, very electrically active, what they tend to do is they spew out a lot of the ion potassium. And that's called K+ also. So potassium then accumulates in the extracellular space. But this is a bit dangerous because if too much potassium accumulates in that extrasynaptic space, extracellular space, the neurons actually start to become even more active, and you can get runaway activity, and you can, for example, get a seizure. And so what the astrocytes normally do is they take up excesses of potassium, they distribute it through their network, and in the end they can actually dump excess potassium around blood vessels so that it's safely transported away from the neurons. And the same happens at these spots where the oligodendrocytes wrap the neurons in myelin. So that's also a place where a lot of potassium tends to accumulate under the myelin. And
Frozen again.
Yes.
And maybe I should say that these end feet of the astrocytes are really crucial in that process. Am I gone again? No.
No, you're here. You were shortly, but
Oh,
not for a long time.
okay. Yeah, I'm really sorry. Normally this goes well, but today is a special day maybe. So when we try to look then where specifically we find MLC1 or GlialChem, I said already we find them in these astrocytes, but when we stain and we look in the brain in detail, we find actually that these proteins are present around the blood vessels in these end feet. So here you can see brown staining for MLC1, and this is a blood vessel. You see the surrounding of the blood vessel, and here also in this image is very much colored brown. In an MLC patient that of course doesn't have MLC1, we find of course no MLC1 surrounding the blood vessel. And here in these pictures you also see a blood vessel. In this case MLC1 is colored red, GlialChem is colored green, and you see that both of these proteins are present surrounding the blood vessels. Now if we look in the brain of an MLC patient at what is going wrong with the cells, we find actually that now we zoom in even more on the blood vessel. So here you see the circumference of a blood vessel. There are some blood cells inside. And in light gray here, you see one of these end.
Ooh.
It shows that these structures tend to swell up. If we then look at the myelin sheets in MLC, we find that there also the myelin sheets contain a lot of these fluid-filled vacuoles. So there seem to be all these bubbles with fluid inside them in the myelin because it seems to have burst. So it seems as if,
Rogier, sometimes you are gone. So would it be better to close your video perhaps? Sometimes
can
that
do that.
helps.
Yeah, I can do that. Yeah. Let me check where I can do that. Okay. Yeah, let's hope that helps. And if it keeps on happening, let me know. So as I said, because of the disbalance of ions and water in the brain, we think that these astrocyte end feet tend to swell up and the myelin sheets tend to swell up, the places where the potassium flow is happening. Now, why would that be the case? And for that, I'm going to briefly talk about what can cause swelling. And so what I said is that when neurons are very active, they tend to spew out lots of potassium into this extracellular fluid. So here I've made this neuron very active. I've only shown potassium ions in this drawing and chloride ions. The others are of course there, but for simplification, I've left them out. So you see accumulation of potassium when there's a lot of neuronal activity, which potentially is dangerous. So what the astrocyte does is with pumps, it can pump in this excess potassium and take it up. But because the astrocyte then takes up extra salts, water is also pulled into the cell by a process called osmosis. And that causes the astrocyte to swell. Now, swelling, of course, is not a pleasant experience for any cell in our body. And so all the cells in our body actually innately have a mechanism to deal with cell swelling, which is called volume regulation. And crucial in that volume regulation is that cells then open specific ion channels. So these are channels in the outside of the cell which can allow flow of these particles again. And these specific ion channels, one of them that plays a crucial role is called the volume regulated anion channel. And so this, or FRAC. And so this FRAC opens when the cell swells up. And because it is a channel that allows, for example, chloride to flow through, chloride flows out of the cell again. And with the chloride flowing out of the cell, water again is pulled out of the cell by osmosis and the cell recovers its original volume. And so the take-up of potassium causes swelling of cells, but then the channel that leads chloride to flow out again restores the volume. Now, about 10 years ago, the lab of and we are looking at the size of these blood cells, and then we give them a shock that causes swelling of these cells. And you see the cells swelling, and then in the time period that follows, you see that the cells are slowly recovering their volume back to normal. And if we make a graph of this, you can see here the cell has its original volume. We give the shock, the cell swells, and then in the half hour that follows, you see the cell slowly recovers its original volume. If we do the same experiment in cells from an MLC patient, you see that this process of volume regulation is disrupted. So the cells are not able to recover from swelling that well. And so regulatory volume decrease is disrupted in cells from MLC patients. And I don't show that here, but it was shown that this is because the volume regulated anion channels do not open properly. Now, you can of course study this in patient cells, so that has been done already. But what we further on did is we used mouse models for MLC to study in more detail what is happening in the brain in MLC. And so we have 2 mouse models that were generated in the lab and that were quite extensively validated. And these are the MLC1 null mouse and the GlialCAM null mouse. So these are mice that are not having a healthy copy of the MLC1 gene or not having a healthy copy of the GlialCAM gene. And they actually perfectly mimic what we see in patients. There's increased brain water content, swelling of the astrocyte end feet, and there is this progressive vacuoles appearing in the white matter. And using these mice, we have also shown that this process of volume regulation is defective in astrocytes in MLC. So not just in blood cells, also in astrocytes. And with that, we come to the part where the three researchers in the lab are going to talk about their studies into further understanding what is happening in the MLC brain. And the first is Quinty. So Quinty, take it away.
Yes. Thank you, Dr. Min. So I'm a PhD student in the lab of Dr. Min, and I'm going to give you a brief overview of my project. So, as Dr. Min mentioned before, FRACs are not properly working in MLC. So my main project is ion channel dysfunction in isolated cells in MLC, and I really want to look at why FRACs are not functioning in MLC and if we can fix this. On the right, you can see a nice figure of a FRAC channel and how it is working. So as Dr. Min talked about before, FRAC is important in transporting chloride ions from the inside of the cell to the outside of the cell. In MLC, this is not working as it should be. FRACs are not opening properly, which means that there's less chloride current that goes outside of the cell, or it is not going out of the cell at all, which means that the volume decrease that should happen is not happening anymore in that matter. And this is an important feature in MLC. As you can see in the picture on the left, you can see ClioChem, MLC1, and FRAC. And there is some kind of connection between those two, because we know in MLC that ClioChem and MLC1 are not working properly, and also that FRAC channels are not working properly. So there's some kind of connection, and this connection is something we really want to explore. So my main question is, why are FRACs not functioning in MLC, and is there some kind of way we can fix this? Next slide, please. So we want to study these FRACs in MLC, but how can we do that? For this, I'm using an astrocyte cell culture to study this ion channel dysfunction in MLC. And for this, I use an MLC mouse model. This MLC mouse model, these mice have a disease that really resembles MLC. So this is a very easy method to really look at how MLC works by using this mouse model. Next slide, please. So what can we do with this mouse model? As shown before, astrocytes are very important in MLC because they're important in the volume regulation, and FRACs are also present besides MLC1 and ClioChem in these astrocytes. So we take astrocytes out of the MLC mouse, and we've grown them up into a culture. And in this way, we can use astrocytes to study FRAC currents. Next slide, please. So we then have these astrocytes in culture. And what can we do with these astrocytes to really look at FRAC and how it is working? We can do like different types of experiments. One of the experiments is staining, as you can see in the black picture on the screen. And staining, this means that you can give different elements in the cell different colors. So one of the colors is blue, and that's DAPI. And DAPI gives the DNA a blue color. Then the second color is red, and this resembles, this is the coloring of F-actin. Actin is the cytoskeleton of the cell. And like every human being, everything needs some kind of skeletal structure. And this structure gives the cell form, like your skeleton gives your body form. But a more important experiment in this, for this, is electrophysiology experiments. And I will be talking about this a little bit more in my next slide. Next slide, please. So we are doing currently electrophysiology experiments in the MLC mouse model. What is electrophysiology? With electrophysiology, you can study electrical properties of the cell. And in these astrocytes, we do patch lamp recordings. Patch lamp recordings is when you have a glass pipette, as you can see in the picture on the top. This glass pipette, you can attach it to the cell. And with this pipette, you can really measure how much chloride goes out of the cell. And this is, of course, what should happen in a normal astrocyte. But for this to happen, we first have to make the cell swell. And we do this by putting the cell in a hypertonic solution. And this will cause the cells to swell. And because of the swelling, FRACs will be activated, and chloride will go out of the cell water will go out of the cell, and then the cells will shrink back again. But this is of course not properly, this is not working MLC. So with this method, we can really look at how fracs are doing in this experiment. So the main question was, why are fracs not functioning in MLC astrocytes? And with this experiment, we can really compare normal healthy astrocytes and MLC astrocytes. As you can see in the graph, the normal straight line is how a normal healthy astrocyte should have as is frac working. So when you look at the line, when the cells are swelling, the line goes up, so chloride goes out of the cell, and after a while it goes down. But in an MLC astrocyte, as you can see as a dotted line, the line stays down, which means that there is no chloride current going out of the cell, and which means that fracs are not working. So with this method, we can really study it. And another very important thing is if we can fix this frac dysfunction in MLC. And with this method, we can really use different drugs to see if we can get the dotted line back up to how the straight line is working, to see if we can restore the frac function. And with this, we can maybe see if there's some kind of drugs that can use to make the MLC astrocytes healthy again. And this will be a great therapeutic approach in MLC. So next up, Emma is going to talk about her project.
Thank you, Quinty, and good day to everybody listening. I'm going to be talking about something slightly different from what Rogier and Quinty have been talking about, but I'll walk you through it. So I study molecular signaling pathways. And maybe you remember the image on the left from Rogier's presentation, depicting a cell membrane with the differently colored channels inside that cell membrane. And this membrane with these channels connects the inside of a cell to the outside of the cell, which is called the intracellular space on the inside and the extracellular space on the outside. And in this way, the cell can actually, in a sort of way, communicate what's on the inside of the cell towards what's on the outside of the cell. But there is actually different mechanisms in which a cell can communicate these types of things. And I study a different way. So on the right side of this slide, you see an image depicting again a cell, which is this circle with yellow on the inside. So the yellow is also the intracellular space, and white is the extracellular space. So everything around this is extracellular. And what we see in the membrane, in the circle, is a blue receptor. And this receptor is a different type of protein which is embedded in the membrane, connecting the inside of the cell to the outside of the cell. And the way it does this is via the 3 steps that we see listed. So this receptor can perceive a signal from the outside world, and this can be almost anything. So it can be if there are cells outside of its own cell, if there is heat or cold on the outside, you can even taste sweetness or sourness with these types of receptors. Once such a receptor receives such a signal from the outside world, it starts an intracellular signal transduction pathway. And this is what I study. So this intracellular signal transduction pathway is basically a whole bunch of different proteins talking to each other, connecting to each other, messaging what's going on the outside of the cell. And this can lead to a cellular response. So the cell can actually act upon what's going on the outside once it has signaled it. Next slide, please. So this process we can study in an experimental setting, and this is what I do. So what's obviously very interesting, or sorry, not obviously, but what's interesting for us about this process is that we do not actually know what's going on in MLC. So we know very little about signaling pathways in MLC. And what's more is that a lot of the drugs that are out there on the market right now are actually affecting these types of receptors and signaling pathways. So if we know a little bit more about what's going on in MLC, then maybe in the coming years, we can invent therapeutic strategies. So that's why we study it. So how we study this is I make a system in the lab where I have a cell. So here again on the left is a cell, and the inside is again beigey yellow. In the top corner, we see a brown molecule, MLC, which is embedded in the membrane. And what we have done is we actually kind of hijack this system of the cell so that once there is a signaling cascade, here depicted is NF-kappa B, which is a signaling protein, this cell is going to make a green substance, and the cell is actually going to turn green, which we can see and also which we can measure. So if we go to the next slide, we can actually see that, for instance, if we in this system also put a water channel in the membrane, which is the blue channel next to MLC1, we can see maybe an increase in this signaling protein, NF-kappa B, which then would lead to more of this green substance being present, so the cells become more and more green. So this would give us an idea of, well, okay, MLC1 and this water channel together, they signal via this signaling protein NF-kappa B. That's really interesting. Would it actually also signal via another protein? So next slide, please. If we look then at another protein, for instance, ACT, and we do not see this increase in green protein, in greenness of the cells, then we know, okay, so they're doing something with NF-kappa B, but not with ACT. And in this way, asking all of these questions and putting them in this experimental system, and we can puzzle together what's going on in MLC with MLC1. And hopefully, at some point, we will know what signaling cascades, what signaling pathways are involved in MLC, and maybe we can alter them, ultimately leading, hopefully, to a therapy. So with that, I will leave you, and I'll give the floor to Sven Kerst.
Thank you. Next slide, please. So I'm Sven Kerst, also a PhD student in the Pool of Rogier Min. And as my colleagues already mentioned, a lot of research that has been done and is still going on that has given us many important insights into the disturbed physiology of the astrocytes have been done in isolated cells. However, it is very important to note that isolated astrocytes lack some very important features that astrocytes in the intact brain tissue have. So on the left, you can see this comparison between an isolated astrocyte and an astrocyte present in the brain tissue. And yeah, what's really apparent is that these astrocytes in culture lack these processes, but also the end feed that are so important in the water and ion homeostasis, and also where these proteins expressed by the genes in which we find mutations are localized. So the aim of my project is to study how astrocytes in MLC are affected in the whole brain, so in the intact brain. And like Quinty, I am studying this by electrical recordings, called patch lamp recordings. And I do this in brain slices from a validated MLC mouse model. Next slide, please. So here I make an analogy between astrocytes and a rubber research glove. And so like a glove that can be inflated, an astrocyte also has some tension on the membrane, and under normal conditions, under healthy conditions, this astrocyte, or in this case, this glove, looks like a normal hand, and it has a specific size. And in MLC, we see in fixated tissue, so tissue that is not alive anymore from mouse models, that the astrocytes appear to be bigger. However, we are not sure whether these astrocytes are then just have a larger size or whether they are swollen. And with these electrical recordings, we can record a parameter that is actually proportional to the size of these astrocytes. What we have found is that these astrocytes are not actually larger, so they don't adapt their size to this swelling, but they are just swollen. So the size of the glob remains the same, but it is constantly swollen. And this is a very important finding. And of course, with this parameter, we cannot tell where this swelling actually precisely occurs. So for that, we are also using very advanced microscopy techniques in which we can really image where the swelling in intact living brain tissue occurs. And an example of such an astrocyte that is still alive in brain tissue can be seen on the right. Next slide, please. So apart from this parameter that tells something about the size of the astrocytes, we are also interested in other basic electrophysiological properties of these astrocytes that might give us clues to why these astrocytes have a disturbed ion homeostasis. And what we can also report, and what we have found, is that in MLC, on the right, in the right globe, we see that these astrocytes have a relatively higher concentration of positive ions. So on the right, the most right, you see an arbitrary scale going from more negative to more positive concentration of these positive ions. And what we see in MLC is that this is increased. And like Rogier already mentioned, these astrocytes are very important for taking up increased levels of extracellular potassium ions. And since these astrocytes in MLC already have an increased concentration of these potassium ions, it's very important to see if that affects their ability to take up these increased levels of extracellular potassium. So that's an experiment that we are also currently performing and might give us more insight into how this affects the uptake of and redistribution of potassium. So this next slide, please. So this uptake of potassium is not only, or the ability of this uptake is not only determined by the ability of the individual astrocyte, but also by the network that these astrocytes consist of. And these networks of astrocytes are formed by special connections, and these connections we call gap junctions. And like Rogier already mentioned, these gap junctions allow the redistribution of this potassium and other ions, and to maintain healthy levels of extracellular potassium. So in a healthy network, it might also happen that upon increased levels of extracellular potassium, a single astrocyte or some astrocytes take up this potassium and swell. But in this healthy brain, this network or this astrocyte that is swelling up is able to give this potassium to the surrounding and connected astrocytes. And we are also currently trying to find out whether this network is also disrupted in MLC. So besides from the fact that these astrocytes are permanently swollen, probably, and also less able to take up potassium by themselves, also the network might be disrupted. So maybe it consists of less astrocytes, or maybe the network is smaller or spans a smaller region. So to be able to test this research question, we can load individual astrocytes with a specific dye that is able, like the potassium ions, to flow throughout the network. And thereby we can really visualize this network that the astrocytes make with each other. As you can see in the right image of a slice in which a single astrocyte was first filled with this dye, and after which the dye was spread through the network. And this is another very important experiment, and also might give us some idea of why the astrocyte and why this potassium homeostasis is disrupted in MLC. Next slide, please. Or I think it was my part. Yeah.
Okay, thank you very much. And with that, I would like to come back to the last part of the presentation. Because of the hiccups that we had, I think I am going to go a little bit faster through it so that we have enough time for discussion at the end. And so what I said I would do is talk about therapy for MLC. And because MLC is a heritable disease, genetic disease, of course, what comes to mind very quickly is would gene therapy be an option for MLC? And gene therapy would depending on the disease or the patient characteristics, involve either inserting a healthy copy of the damaged gene, repressing the activity of a disease-causing damaged gene, such as, for example, in the case of dominant GLIOCAM mutations, or repair of the damaged gene. Now, interestingly, last year, a nice study was published by researchers from Spain, where they tried gene therapy in a mouse model for MLC. And so these were MLC1 lacking mice, where they put back a healthy copy of the MLC1 gene, by viral transduction. So a virus containing a copy of the healthy gene was injected into the brains of these mice, and the astrocytes were then making healthy gene product again. And what the researchers found is that actually such a treatment was very effective in reverting the vacuoles in a specific brain region where they actually injected their virus. And this was now, first of all, of course, the genetic material has to be delivered to the brain. And this could potentially, if you use a virus, for example, lead to a dangerous activation of the immune system. Second of all, if you want to get such a gene product into the brain, it's quite an invasive procedure where intracranial surgery is required. However, in the future, maybe it might be possible to put such gene therapy through injection of viral particles into the blood. But then one of the biggest caveats is that which brain regions to target and which cells to target, and how to make sure that you can target all of them. And so in the study that I showed you in mice in the previous slide, only part of the cerebellum was targeted and recovered. But the problems in MLC are, of course, brain-wide. And so you would like to target astrocytes throughout the brain. But because the human brain is very big, this would require quite an extensive amount of viral injections. And at this point, I don't think that's feasible. But I must say that developments here are going quite fast. And finally, there is, of course, always the risk of off-target effects if you start to do intervention with genes. So the integration of the gene or gene editing might lead to new unexpected genetic damage, which could, for example, lead to cancer. So although gene therapy, I think, is on the rise for a lot of different diseases, our group is currently not working on gene therapy for MLC. What we hope to achieve is a pharmaceutical treatment for MLC. And so, as I hope became clear during the talks here, is we hope that we can treat MLC by repairing the cellular processes that are defective, hopefully with specific drugs. And so the primary targets that we have is we would like to repair volume regulation in astrocytes, and we would like to restore the function of the ion channels that are involved in this process of volume regulation. Now, of course, that comes with a lot of challenges, and we need to know a lot about the disease. And I hope that the three talks by Quinty, Emma, and Sven made clear that we are trying to find out why channels are not functioning in MLC, what the primary defect is. That was Quinty's project. We're trying to find pathways with which we can restore such processes by understanding defective signaling in MLC. as Emma talked about. And if we would reach that point, we hope that in intact brains in our MLC mice, using the approaches that Sven described, we can see whether we can restore processes and recover brain function, at least in mice, and then hopefully in the future in humans. And as a wrap up for my talk, I would like to say something about how you might be able to help us as patient families. And so I think one of the things, so we do research on MLC because we really hope that we can one day find a cure for this disease. As families, I think it would really help if you get organized. So through Facebook, through websites, or to organizations like the ULF or the European Leukodystrophy Association. These are great platforms to organize as patient families. They offer the possibility to exchange information, stories, experiences, exchange tips, but also for us in the future, if we would want to reach a large group of patients for potential clinical studies, it is a great Am I still
Yeah,
audible?
you're back. And Rogier, you were with the third point. Help us reach you for potential clinical studies. That's where you were.
Oh, okay, yeah, sorry. So then I was going into the fundraising. So what I wanted to say is that the research that we do is quite expensive, of course. There's a handful of labs worldwide that focus on MLC, but we depend on external funding. And for rare diseases, funding is always a bit limited. And so every little bit of research funding helps. So organizations like the ELA or the United Leukodystrophy Foundation fund such research, but also fundraising by patient families can help researchers to study such rare diseases like MS. And so this is, I think, where you can help. With that, I would like to thank you all for your attention. I would like to acknowledge all the people who have done all the work on MLC in the past decades. At the moment, our group is the three people that you heard talking, and then Mohit Dubey and Elke Brouwers were two previous PhD students, recent. We collaborate very extensively with Mario van der Knaap at the Amsterdam UMC on MLC, and with several other people there in the Amsterdam Leukodystrophy Center. And at the Vrije Universiteit in Amsterdam, we collaborate with the lab of Professor Hubert Mansfelder, who is a neurophysiologist. And I would like to sincerely thank all patients and their families for their help in this research. And then I think we should move to potential questions from the audience. So I'm gonna end my slideshow, stop sharing my screen, and see whether we have questions in the chat.
Yeah, there's a question for you, Rogier.
Yep, I see the question. So somebody in the chat is asking, thank you to all four for a very informative talk. We really appreciate your efforts on research on this disorder. Dr. Min, I believe on Thursday you mentioned that status epilepticus was fairly common in MLC. Is this correct? Yeah, this is correct. So it is well, numbers are maybe better given by Mario, but it is seen in patients more often than in the general epilepsy population.
Yeah, and what's correct also is when it starts, it's more likely to develop into status epilepticus than in standard epilepsy patients. So Marla, thank you for your question. It's correct that I would suggest having something to end the seizure at home. That would be a good thing.
Yeah. Yeah, and so indeed there are rare cases where the status epilepticus has turned out fatal. Yeah, thank you for your question, Marla. Are there other questions from the audience? Feel free to ask them in the chat or to also just unmute and ask them. I think this is also perfectly fine. Somebody is asking, is there a way that we can directly, it's also Marla, that we can directly fund your research? I think that Mario would be the person that that has done that in the past for other leukodystrophies.
We have at the University of the VU Medical Center, the Amsterdam University Medical Center, we have a foundation through which donations can be made. So, which also I think makes them tax deductible because it's a foundation. So the answer is yes, and you can donate with the specification that you wanted to go to the lab of Dr. Min. So Marla, I know that you have my email address. If you email me, then I'll connect you with Dr. Min and also let you know how donations can be made. I think that Dr. Min made an important point because I feel like also from Amsterdam we are a little bit failing on the MLC community, and so we have not instigated you very much to organize as an MLC community of family patients and families. But that's something that we should really discuss. And I was talking to Bob yesterday, Bob Rauner, and because there should have been an MLC session last year, but this meeting was cancelled for obvious reasons. And if we can meet in person next year, he made me the promise yesterday that there will be an MLC meeting, specifically like a half a day. And I think that would be perfect also to try to get as many MLC families as possible and also discuss how you can organize yourself through a Facebook group and share information and also share questions to researchers. And if you would talk to Donna is doing the same thing for Vanishing White Matter. Donna Squirrett, she is on the board of the ULF, and she might help you and using Vanishing White Matter as a model. So that would be helpful, I think.
Yeah, and for us as fundamental researchers, not as clinical, because Mario, of course, is a clinician and sees MLC patients very often. But for me, as a fundamental researcher, I think such a ULF meeting would be also a great opportunity indeed to get more directly in contact with patients and families and to
and
start
get to know each
dialogue
other.
and get to know
Yeah.
each other indeed. Yeah.
And connect.
Yeah. Are there additional questions? I think we have about 2 minutes left before the Zoom meeting has to stop. Or remarks or things like that. If not, then I think I will wrap up, and I would so I hope that we've together been able to give you a feel of the kind of research that we are doing in the lab in Amsterdam. As I said, there are several labs in the world that work on MLC that are actually the interesting part is they are all taking a slightly different approach towards understanding the disease, which I think is very valuable. And together we really hope to understand what is going on in this disease so that we can come to a treatment as an ultimate aim. So thank you very much for your presence today, and feel free to contact us by email also. So I should have put my email address in the presentation. I didn't do that, but I think you can find me online, and yeah, feel free to do that. Thank you very much.
And also, if there's any additional questions not addressed in the session, you can email them to office@ulf.org and we can pass them along as well. I want to thank the speakers. They were excellent presentations. And thank you to our sponsors, our audience for making the virtual family conference this year a possibility. And we hope to see you next year in person. So thank you very much.
Without the internet hiccups, hopefully.
Yes. Thank you.
Okay.
Thank you. Thank the ULF for having us.
Thank you very much. Yeah.
Thank you.
Bye-bye.
Bye-bye.
Bye.